VGAS. Visualizing the coverage view. Once the groups in cutoff have been set, we click the Update button to generate the coverage view. Here we have a gene selected, and the sequences we are using have been loaded from 3 BAM files, each containing next generation data for a single subject. For the cutoff, we are using a read percent of 5. The coverage groups are displayed in the Analysis Display panel below the sequences. If we scroll the sequence scroll bar, you can see the sequences will scroll as normal, but the coverage groups stay in place. However, if the coverage view is too large to fit in the area below the sequences, then a message will appear asking you to lock it. To lock the coverage and hide the sequences, we click the padlock button at the bottom left. The sequences are now hidden, and the scroll bar will scroll the coverage groups. The coverages can be locked and unlocked at any time by clicking the padlock. Okay, we're going to hide the gene panel just to give us more screen space. And now we're going to go through the display options. Each row represents a group. Here we have 3 groups, each representing a sample generated from 3 BAM files. The nucleotide consensus, so the top row is the consensus made up of all possible bases found at that position. So we see either an A, C, G or T, or an ambiguous code where more than one base exists at that position. The next row are ordered by base occurrence. Therefore, the second row is the majority consensus. Nucleotide coverage count. This is the same as the consensus, but with the count of each base shown below. Nucleotide coverage percentage is the same, but with the percentages. These can also be displayed for the amino acid coverage. However, no consensus is displayed on the top row. So amino acid coverage count, amino acid coverage consensus. A coverage group can be selected by clicking on it, which is then highlighted in green. This is the same as selecting a row or sequence. Sequence group matches. This highlights the nucleotide bases or amino acids in each group which match the selected group or sequence at that position. Show insertion information. This displays a green line at each position that an insertion exists within the group. The tooltip lists each insertion along with its occurrence. Highlighting group differences. We can highlight the differences between a selected group or by group wide. These are shown as red bars. We can also ignore the occurrence order. The selected group, and the group wide differences also works with the amino acids. If we display the gene panel and select display on gene view, then on the gene panel the group wide nucleotide differences, each base position is shown with a little red line. Average quality scores. Each base will be shaded depending on the average quality score for that particular base at that position. The lighter the shade, the higher the quality score. The tooltip also displays the maximum, minimum and average score at each base. We can also copy the consensus or majority for either the selected group or for all groups to the clipboard, which can then be pasted in faster format. Nucleotide coverage plot. This plots the bases at each position stacked in a sort of bar chart, where both the scale and the plot height can be changed using track bars. So the scale and the plot height. Again, the tooltips give information about each base position. And by clicking on the plot, we can navigate to that position. This can also be used to generate a coverage plot. However, you will need to change the cutoff type to percentage. So this takes into account the unsequenced or empty bases. You can also show or hide the nucleotide colours. Export into an image. The To button expands the form to display the image export options. Here we can set the values that generate the image into various image formats. The image can be generated and it will be exactly the same as whatever's shown within VGAS. So feature wide, or just a partial map. You can include the scale, nucleotide bases, amino acid bases or residues. You can size it for the amino acid scale, which will bring each codon down to one amino acid. And you can also size the actual image, and this gives you an idea of the actual image size. You can also have a transparent background or set the image color, background color.